Hello, everyone. We are just waiting for the attendee list to kind of level off, and then we'll get started. So just another moment. All right, well, it looks like that list has started to level off, so we will go ahead and get started. So welcome to our 2022 ClinGen Clinical Genomics Career Panel Series. Oops. All right, there we go. So just as a reminder, this is a multi-part series. We kicked this off last week with a panel focusing on genetic counseling, and we will continue for the next 3 weeks after this, same time, same place. Our next panel will be focused on physician clinicians, followed by research, development, and other opportunities, and finally, stepping stone opportunities. So jobs that you might consider maybe right after you've graduated from college, for example, before you decide what you would ultimately like to go on to. You may be at this panel today because you are already interested in a career in laboratory genetics, but I encourage you to try to attend the other sessions as well, just in case you hear something that you have not considered previously. All of these will be recorded, and if you didn't catch the genetic counselor one, that recording is available. So want to thank everyone who provided questions for our panelists as part of the registration process. But certainly if something comes up, something that you want to ask while our panelists are speaking, please feel free to type that question into the Q&A box. This is a webinar format, so you're not going to be able to come off mute. So that's going to be the way that you will communicate questions to me, and I will ask them to the panelists. And if you see a question come up that you are also interested in, you can vote for questions that you would like to see addressed. So with that, we'll just do a quick poll. So you should see the first question come up on your screen, and we're just trying to get a sense of who our audience is today. So the first question is, as of September 2022, will you be a student? And it asks you a few choices of what type of student you will be. And then the second question is, how familiar are you with the genetic laboratory career opportunities? So I'll give you a moment to record your choices. And Jules, I don't know if you are able to see the answers coming up. There we go. All right, so just so everyone knows who's on the line with us today, we have some people, about 20% of the audience will be undergraduate students. About 6% or one person will be a master's graduate student. 2 people will be PhD students, but the vast majority of people on the line will not be students as of September 2022. So perhaps individuals who are still actively considering what they'd like to do in the future. And then how familiar are you with the genetic laboratory career opportunities? Most people felt like they're moderately familiar. At least 2 people, well, 4 people total said they were either very or extremely familiar, but we have about 5 people that are either slightly or not very familiar. So a good mix of people on the line with us today. With that, I am actually going to turn it over to my colleague, Stephen Harrison, for a moment to provide you with just a brief background about what clinical laboratory genomics is. So Stephen?
Perfect, yeah. So because we're talking about careers in clinical labs, what defines a clinical lab? So typically this means it's a lab that is performing testing on humans where those results can go into the patient's medical record and potentially impact medical care. In the US, at least, this is regulated by CMS, the Center for Medicare and Medicaid Services. In other countries, this will vary as far as what the regulatory oversight is for what counts as a clinical lab. In order for a clinical lab to function, they have to have a CLIA license, and this helps to promote consistency, quality, and transparency in laboratory testing. and all test results that come from clinical labs in the US, the CLIA number has to appear on that test to kind of, again, help show the, that they've gone through that effort of having a CLIA license. There is approximately over 260,000 CLIA labs in the US, but this is not specific to genetic testing. This is any kind of human specimen testing, microbiology, pathology, tumor testing, and then also, again, including genetics and cytogenomics types labs. So on the next slide, as far as how does this compare to research labs? So again, in more of a clinical setting, you are often doing indication-based testing to see if someone has a family history of disease or disease themselves, to see if you can find an explanation for why they want testing. And these results typically go to back to an ordering provider. But because this is this is patient samples that can impact medical care, there are certain certifications and training that would be required for someone to work in a clinical lab setting versus running the same types of tests, but under a research lab setting. There's also more regulations and rules about SOPs and keeping equipment up to date, and additional sort of restrictions that even though a research lab and a clinical lab may run the same tests, there's additional kind of oversight that goes into it on the clinical lab side. Whereas on the research side, you may be using human specimens for your testing, but the results may not go back to the participant, depending on how that study is set up. But it is important to note that these are not mutually exclusive. A lot of labs that do some clinical laboratory testing also have research efforts as well. To validate new tests, they may work with certain clients who don't need a clinical report, so they want to more do clinical grade research and work with a lab or a hospital in order to implement these changes. So yeah, it's not always an either or when you think about clinical versus research testing. Next slide here. You can see there are lots of different career opportunities within a clinical lab testing environment. Some of them are broken up here a bit by kind of a wet lab versus a dry lab. So in the wet lab is where we think about the physical running of specimens. Sample receipt and accessioning, actually performing the test, but then also all the quality control efforts that go into making sure the test is run properly, maintenance on equipment, different automation factors, and sort of a role that kind of spans the wet and dry is different R&D efforts of, okay, we have this test, we want to add more genes to this test. How do we go about adding that? How do we validate which tests we should be offering? How can we make the testing process better? Is there, should we move to a different sequencing option, a different capture option? And on the dry lab side is more of then kind of the downstream efforts of, once the results come off the sequencer, it has to go through a pipeline. So there's lots of people involved in the bioinformatics process. And then, efforts on the result interpretation part. So we found these variants. We have to assess these variants, figure out the clinical significance. There's also teams involved then in the putting together the report, taking all the different pieces, putting the report together to see, does it make sense? Can we explain part of the phenotype we're seeing in this case? Also then sign out from typically a lab director of then reviewing everything and processing that sample. There's also roles here, for quality management and even some, more clinical research of looking at all the data you've been assessing from tests and seeing if you can educate your own processes of like, how often do we find variants in this gene with this phenotype, and being able to sort of, look at your own data and see if you can make any, conclusions and improve your processes with this information. And then on our next slide here, you can see that also there's sort of a range of disease areas that you could be working in. A lot of labs will span different disciplines, like cancer versus cardiology, or rare disease is kind of often a bucket that a lot of testing falls into, or even just the actual, being involved in the testing process itself of, more of the wet lab side. And on the clinical lab side, it also, can vary where you're working. Universities have clinical labs, commercial clinical laboratories, hospitals, nonprofit institutions. There's lots of different settings in which you can work in a clinical lab under any kind of these sort of umbrella programs to be able to get some insight into how these all work. I think that overall. Oh, and then some additional resources if you're interested, in different training opportunities. Some links here to the ABMGG, the American Board of Medical Genetics Genomics, National Society of Genetic Counselors, NSGC, ACMG, and then also AMP in the pathology side. So a lot of these have different career opportunities. Perfect, looks like Jules put it into the chat as well, to sort of learn more about some of the different paths you could go that could lead you to working in a clinical laboratory.
All right, well, thank you for that brief overview. That's just kind of to set the stage so we all have at least a basic idea of what we're talking about when we're referring to laboratory. And with that, I will kick off the panel. So the way that we'll start out, I will ask each of our panelists as displayed on the screen to introduce themselves. So when I call your name, please feel free to say who you are, where you work, what do you do, if there's any sort of special degree or training that was required for your position. And as we rotate through the 4 panelists, if someone has already said something similar to your own career path or something that someone else has already shared, just feel free to acknowledge that you took a similar path rather than reiterating the same story if that's applicable to you. So with that, I'm going to stop sharing so we can see each other's faces. And I'd like to start with Marzia Pasquale.
Hi, thank you for inviting me. My name is Marzia Pasquale, and I have a PhD and a board certification in clinical biochemical genetics through the ABMGG. I am a professor of pathology and pediatrics at the University of Utah, and I'm also the section chief and medical director for biochemical genetics at ARUP laboratories. So I think I have the best of the 2 worlds. I am in academia, but I am also working in a reference laboratory. So I get to do a lot of teaching. I also am a program director of the fellowship in clinical biochemical genetics. So I teach fellows, I teach graduate students and medical students. And also I do clinical service, and the clinical service consists in results interpretation. And the tests that we do in my laboratory are tests that are used to screen, diagnose, and monitor patients with metabolic disorders. So it's a very, I think, a rewarding job, at least for me. Every day and every patient, every test results is different. It's like solving a puzzle. So you need to put together clinical information and test results. Sometimes the puzzles have only 6 or 8 pieces. Sometimes they have 500. But that's what makes the day really interesting. I've been doing this job for a long time, as you can tell by my white hair, and but I still have a lot of fun. I love what I'm doing, and again, I hope that someone will be interested in this career as well.
All right, thank you. Next, I'll move on to Raj Ghosh.
Yeah, hi everybody. Thank you for inviting me. And I am as Raj Ghosh. So I am a board certified laboratory genetics and genomics in laboratory genetics and genomics by ABMGG. My background would be PhD in molecular genetics, then doing postdoc in statistical quantitative genetics, and then moving into LGG fellowship, and then getting board certified. And now I currently work on, so as you can say, to probably tell that I spend a lot of time in academia, and then I wanted to do something more applied. And I am basically a testament to what Stephen mentioned, that these are not mutually exclusive. I work at the NIH, the largest clinical research hospital, as in the at the clinical center, which is the largest clinical research hospital, at least in the United States. And here what I do is I sign out exome and genome results of patients who are enrolled at the clinical center. And also at the same time, I have access to data so that I can do research on the data, on the genome data and the exome data, not only to try to identify new diagnoses, but also try to improve the technologies. For example, one thing you appreciate, and that's kind of when anyone gets into this field, that it's a really fast evolving field, moving from exome sequencing to genome sequencing, non-long read sequencing. There is RNA-seq, so there are lots of new tools are being developed, and we are trying to make sense of it, how to best use it. And so a lot of my time is also involved in research. So I'm a part of the centralized sequencing program. I'm clinical molecular geneticist at the centralized sequencing program at the NIH, which started from NIAID. So as far as specialization goes, I learned a lot about immunology over the past couple of years, and now it has grown into different programs from different institutes, such as National Institute of Mental Health, stroke, neurological and stroke disorders, and so on and so forth. So you get a wide range of flavor of different types of disorders and their molecular mechanisms underlying them. And at the same time, in this position, you can integrate research as well as sign out patients like a lab director would do in other settings.
All right, thank you. Carolyn Maines.
Hi, everyone. Can you hear me okay? Okay. So I'm a cytogenetic technologist at the University of North Carolina, and I've been here for 20 years as a technologist, and I more recently moved into the role of supervisor. So I'm one of the people that's more doing a lot of the wet work or bench work. So cytogenetics is the study of chromosomes and looking at and diagnosing that syndromes such as Down syndrome, and then also a lot of cancers. I'd say 80% of our workload is cancers. So looking for the Philadelphia chromosome, CML, stuff like that, leukemias and lymphomas, and following them, the patient, for residual disease. So we do a lot of microscope work, fluorescence, FISH technology, and also we do, for the DNA level, we do look at microarrays. So there's a lot of variation in cytogenetics, which is nice. And I did my education mostly in Canada in a genetics technology program before moving to the States 20 years ago, where I later did my master's in molecular genetics. So I am dual certified to work either in a molecular lab or a cytogenetics lab as a technologist. So if you have any questions about that, I can try and answer them.
All right, thank you. And Stephen Harrison.
I'm Stephen Harrison. I'm a laboratory director at Ambry Genetics. My background or training is similar to Marzia and Raj, and then I did a PhD and then did a ABMGG fellowship. I did mine in clinical molecular genetics, which no longer is an option. It now has merged to the laboratory genetics and genomics program. And I, prior to joining Ambry, worked more on the academic side, and then now have made the move more now onto the commercial clinical testing side, but still work on the ClinGen project and some other research projects on the side. But primarily at Ambry, I do a lot of case sign out. We do a lot of inherited cancer panel testing. So a lot of reviewing the, sort of the phenotypic information we get with what we found from testing and seeing if it, makes sense. Also some cardiology testing, exomes, and some other large panel testing, but then also working with the variant interpretation team that we have to make sure that the process is the same, because with all, with lots of different testing, different people are assessing variants, and you want to make sure that the process everyone goes through is the same, so it's not sort of person dependent, and working with kind of making sure that the decision trees and flowcharts are in a way that, there's consistency. So working a lot more on the sort of variant interpretation side, so that when it gets there for a sign out, you have faith in the classification that's on those variants and are able to review those with confidence. So it's good.
All right. So now that everyone has a sense of who all is on the panel, please feel free to put your questions into the Q&A, and as they come in, I will read them out loud. But I'll just get us started with a general question. So if everyone could say a word about how did you hear about this particular career option? What kind of got you interested in this? What led you to where you are today? And anyone can feel free to shout out.
When I was in grad school, we were running some research exomes, and I enjoyed the having a patient sample and seeing what we could find. At the time, we worked with a lab that was on site. I was at grad school at UT Southwestern, and someone there mentioned this fellowship program where you can, still work with patient samples and do reporting, but you don't have to have an MD. You could be a PhD. So it was about halfway through during grad school when I learned about this career opportunity, and I was like, oh, that's exactly what I want to do, because I enjoy this process of the testing and seeing if I can make sense of the puzzle, but I also don't want to go be an MD geneticist. I like this part, and realizing that there was a process to get there and be able to work in more of a clinical lab setting with patient samples without having to meet with the patients myself, and be able to be involved in this kind of research.
And Carolyn, what about you? How did you hear about the technologist career option?
So I was doing my undergraduate degree in biology at the University of Guelph in Canada, and I was taking a lot of genetics classes and just really enjoyed them. And I met another student whose mom either taught or worked at the Michener Institute in Toronto, which has a specific genetics program to get you into the field as a technologist, either as a molecular technologist or cytogenetics. And of course, I'd never heard of cytogenetics and didn't really learn anything about it until I got into the program and started. And in fact, until I did my clinical rotation, I didn't have much interest in studying these crazy worms with bands that I had to match up. I was more interested in the molecular. So that program was nice in that it gave me flexibility to be able to choose either, but by the end of my clinical rotation, I was in love with chromosomes, and I haven't looked back since.
Raj or Marcia, would you like to add anything?
Yeah, go ahead.
I always liked the chemistry, and in fact, when I was growing up, I was probably 11 or 12, and my parents gave me a chemistry set. I set up a mini lab in the basement. I don't think they were very, very happy, but but I liked the chemistry, and I liked the ability to apply chemistry to medicine as well. So again, this was the perfect marriage.
Yeah, mine is kind of similar to Stephen's, and although it's like it's different in some ways. I had no idea of the clinical genetics as a field when I was doing my PhD. I worked with worms, C. elegans, and then I did. But I like genetics, and my whole career has been through genetics. Then I did the different type of genetics, which is more quantitative. So I picked up some quantitative skills, and it was during that time, it's 2012, when the genome sequencing or exome sequencing was becoming cheaper, so much cheaper, so that it is available as a molecular test. And that there are several papers which I came across, and that made me decide essentially to switch fields. It was kind of a leap of, I mean, I was still working with worms, so it was kind of a big jump. So at that time, I thought that, okay, I have lots of genetics background, I will apply to the fellowships and I'll get in, but it didn't happen that way because I had no human genetics background. So I needed some amount of familiarity with, clinical laboratory tests or human genetics, but I had none of that. So I joined ClinGen at that time. I worked as a staff scientist there for 3 years. which I learned a ton of stuff about. And they're smiling because Erin and Stephen was also there at that time. And Erin is still there, and Stephen is still there, and I am still a little bit a part of it. So once you're in ClinGen, it's a fantastic place if you're trying to make a career to go through the ClinGen process and understand, to get familiar. If you're like me, no background, to get familiar with how the clinical genetic testing works, and that helped me get into the fellowship, and that's how I got in.
We did not pay Raj to say that, just so you know.
Oh, okay.
It's
yeah.
not an ad.
Oh, yeah, that's true.
But we now have a couple questions in the chat, and one of them kind of segues into what you were just talking about, Raj, and we'll see if you have any other comments or if someone else wants to comment. But someone's asking, how much experience in genomics, oops, it moved on me. How much experience in genomics do you need to be able to get into this field? So if someone is a biology and environmental science double major, could they still get a job in the field with limited experience? And Raj, you kind of started talking about that, but would you add anything else?
So it depends on what at what level you're trying to enter. If you're trying to become your goal is to become a laboratory director, you have to get into a fellowship. So that is very competitive these days than it was even a few years ago. So given that competition, I would think familiarity with some level of clinical genetics tests or human genetics, that would definitely help.
And there are some related questions specifically about what do you need to get into the LGG fellowship? What could people doing their PhDs do to prepare themselves, or any other specific requirements that you would point out?
Unfortunately, it is very competitive. As Raj said, that it's sometimes honestly just emailing. If let's say you're midway through your PhD or grad school and eventually this is where you want to go, I would start reaching out to some of the programs now and saying, what could I do to be a good candidate a few years from now? Because some of the programs will vary a little bit. But even just kind of getting yourself on their radar and showing that initiative of like, this is what I want to go do. What can I be working on? Because at the end of the day, they do get a lot of applications, and that they're kind of have to sort of go through, because it's, they could get maybe anywhere between 30 to 100 applications for one spot. And so the reviewers kind of go through things, I think, relatively quickly. That having anything to be able to sort of like call out and make yourself different, to be able to sort of, talk about any experience you have. And it doesn't have to be in a clinical necessarily laboratory setting, of just sort of any work you've done in your, grad school programs, any, kind of where your interests are, what do you think about the field, something to sort of help you stand out a bit more. But again, I do think reaching out to labs is a good option, because unfortunately, when the time came to apply to places, I was like, oh, there's like 30 to 40 programs. This will be great. I can apply to all of these. And then as you start looking at the programs, you'll realize some of the programs only take someone every other year. Some programs will only take people who have their own funding and they won't fund you. Sometimes also places have sort of a internal wait list where there's people who are already working there that, are working there on a postdoc and they're planning to start in 2 years. And so sometimes that's why I would recommend reaching out to the lab, because it could be that they say, we often don't take people straight out of grad school. We want you to come work as a variant scientist first, or to have these other opportunities. I think being able to get some of those under your belt will then help when the time comes to actually apply.
If I can add something, and again, biochemical genetics is a little bit different than the LGG, and it's probably more clinically and patient oriented than the other two, than the LGG. So, but for both, I think that not just reaching out to the laboratory, but if you can have some experience, if you have a genetics program and you can participate in some of their case management rounds, just to have an idea, because clinical service, especially in clinical genetics, is very different than research. Most PhD students have experience in a research setting, and the clinical setting is very different. And again, especially with biochemical genetics, having some opportunity to be exposed to a metabolic service, perhaps attend some rounds or even clinic if they allow observer. That would be very, very good to really understand, is this the career that I like, that I want to have?
And Carolyn, do you want to make any comment? I know we've heard a little bit about supply and demand for the fellowships, but what about in the technologist career space? Are there many opportunities, or is that more of a competitive thing as well?
There are a lot of jobs and a very high demand for technologists right now. I know a lot of labs that are struggling, and there's a huge conversation among a lot of the directors as well on how to fix this shortage. So I know a lot of labs, or a lot of commercial labs, take in people with a biology degree. So I think even if you're interested in an LGG, I've known people who've applied for like senior tech positions or kind of other lab technologist roles and moved on to become a LGG fellow and director later on. So there's that path as well. And then it's just a technologist too. You can get into the field a little easier through a large reference lab. And then if you want to get into a clinical lab or like a hospital academic setting, it's easier to have some experience from a reference lab than to get into a smaller lab. That way, I don't know if that answers the question, but.
Okay. Now let's talk about other ways that you can gain experience. Somebody brought up variant scientists. Does anyone want to comment on the roles that variant scientists play in the lab? What kind of degrees people who are variant scientists tend to have, and can that translate into one of these other careers potentially?
Yeah, I think that definitely variant science is a great a good path if you're interested in this field, whether you want to go into a fellowship someday or stay within variant sciences. The term variant scientist is a bit vague, and labs may use those differently. I know there are some labs that would hire a variant scientist that has a bachelor's, and I know some that would hire people that have a PhD to be a variant scientist. So it is there's not a specific credentialing that is needed to be a variant scientist. It's sort of up to each lab, I think, how they implement those. But that is a really great way if you're interested in this field of sort of getting in, because sometimes they can even be remote positions. So it sometimes could be a little more easy to get into a place if you're willing to be remote versus on site. But it sort of gives you a good insight into the clinical genetics realm of realizing the standard when it's clinical versus research is much different. That it's can't be like, oh well, this predictor said it's pathogenic, so it must be it. That like there's a lot more that goes into the whole decision-making process, and kind of even just understanding the workflow of the lab of it's how we found this variant, then it goes to the variant scientist, then it goes to the person doing the report, then it goes to sign out, and kind of just learning about the whole structure. That is a good way to get it. That's how I started. I had applied to fellowships when I was in grad school. Unfortunately, didn't get into any at that point. Then worked as a variant scientist for 2 years, and then was able to go move into a fellowship that was at the same place where I was working as a variant scientist. So I can say, at least from my experience, that worked. It was a good process to get in, and was very helpful to sort of learn it a bit more on the ground floor of kind of how it works before then moving into more training.
Thank you. So let's switch gears a little bit, and I will ask a different question coming from the audience. Can you talk about the role of bioinformatics in your careers? Obviously, we're all impacted by it in some way, shape, or form, but do you yourself feel like you need to know how to program or anything like that? Does anyone want to take a stab at that one?
Yeah, I mean, it depends on what you're working on. So if you are in some kind of a hybrid situation, like in a job like at NIH, so you have access to a huge amount of data, genomic data, and definitely bioinformatics skills help. So there are multiple levels. I mean, you could just know enough bioinformatics to just do some basic data analysis. That definitely helps, because you'll be working, if you are interested in research without the wet lab part, you would have to mine the data, and some programming skills definitely helps, and that will help you stand out in the future job. If you are applying for jobs like where there is a hybrid, there's a need for data analysis that feeds back into the actually clinical reporting as well in the future. And the other thing that helps as a lab director is that you also can converse with bioinformaticians more effectively in developing the pipeline so that it suits your kind of what you need. So because bioinformaticians are not trained in clinical genetics at all, they are very well trained, in my experience, in implementing algorithms, and they can implement something you want, but you have to convey to them what you want, and it's good to have a sense of what's plausible and what's not, and some familiarity with the sequencing process definitely helps. And that also lets you understand what are in some cases, what are artifacts and what are not. And then you can go back to the bioinformatics team to look a little bit closely and so on. So it definitely helps.
Any other comments on that one? All right. Do each of you want to talk about, you all mentioned you are boarded in something. So can you talk a little bit about that? What is the process for becoming recertified? Do you need to retake exams? Is it just continuing education kind of things? Can each of you speak a bit about that?
I can speak for the clinical biochemical genetics fellowship. So in order to become board certified in clinical biochemical genetics, you need to complete a 2-year fellowship in an accredited training program. During these 2 years, you learn a lot of genetics, medical genetics. Your focus is going to be in clinical biochemical genetics, learning all the methodology and the tests that are used in a clinical biochemical genetics lab. You need to have hands-on experience, and you need to, at the end of 2 years, have completed the logbook with 150 cases that comprise pretty much the whole experience in biochemical genetics. The board exam is composed of 2 parts. One is general genetics, which includes medical genetics, so dysmorphology, embryology, cancer genetics, everything genetics, has some molecular cytogenetics and clinical biochemical genetics question, but very few. And then a specialty exam, and this is focused on your area of specialty. So it could be LGG or it could be clinical biochemical genetics. I've seen a question like, how many times do you have to take it? Hopefully only once. That's, yeah. I think the majority of the applicants, they take it once. You can take it sometimes a couple of times, but I think it's a hard exam. I don't want to say I think it's hard, but I think if you study, you can pass it. The board exam is given every 2 years. And so you have the opportunity to take it every 2 years, but it's taken, you need to pass both exam in order to be board certified. Once you are board certified, if you want to take another specialty, then you only take the specialty exam. The general exam, you don't need to take it again, but you need to be board certified.
And once you're certified, how long is that good for?
Well, right now we are, there is a continuing certification process, and you have certain activities that you need to do in order to be considered continuing to participate, to be active in your continuing certification. And the cycle are 3 years a cycle right now. So there are projects, well, there are questions that you need to answer every 6 months. There is a series of questions that are really related to your specialty. There are quality improvement projects that you need to do. There are a lot of other activities that you need to do in order to be considered active in this process.
And what you just described, I believe it is very, very similar for LGG, right, Stephen and Raj? So I might just pass it on to Carolyn to see if there's anything different about the certifications you have, what you had to do to get those.
So once you're a technologist, and a lot of labs require you to have your ASCP certification in the field you're working in. So I have it in cytogenetics and molecular genetics, and that's good for 3 years. So you take a test, and a lot of labs will give you, if it's contingent on your employment, if it's required by the lab, you have about a year to take and pass that certification. And then it's good for 3 years, and you have to do similar things. So continuing maintenance, continuing education talks and stuff like that, to be able to just recertify every 3 years.
Thank you. All right, another question coming from the audience. This is always a good question. What do you enjoy most about your work? And then what do you find most challenging about your work? And we'll ask everybody to comment on that. I know you have something.
I say mine might, I mean, it's maybe the same answer for both. The more interesting cases, the harder cases, those are the harder one. I mean, they make things more interesting because, you know, when it's just a straightforward BUS, it's relatively straightforward, but it's those more complex cases where like there's something weird going on here, where you spend more time kind of learning about that disease and the different phenotypes and the variant spectrum and kind of putting the whole thing together. I say those ones are also sometimes hard because sometimes you're looking at a case, you're like, I know there is something genetic here. They have this really strong family history. We're not finding anything. And like, how long do you keep digging to be like, maybe go look in all the introns. Was there anything here? Was there maybe a really low level somatic variant? And so the kind of knowing when you have to just sort of stop because, there isn't anything there versus, how much you keep digging and let me like change this allele frequency threshold to see if there's anything else that could be here and kind of keep playing around to try to find something is, can be difficult. But again, those are the interesting cases when there's just such a strong, genetic component to that case that you can tell there's something there that keeps it interesting.
Carolyn, what about you? I see you off mute.
Okay, I'll speak from a technologist point of view, not supervisor, because I enjoy that, the technologist. And being able, what I love most is just being cross-trained in a small laboratory, I could start my day reading G-bands on the scope and finish reading fish on the fluorescence scope, and help out in areas, in the wet areas, or reading a microarray case. So just having the flexibility and the cross-training to do a variety of things in one day even, or across my weeks, I enjoy. What I don't like sometimes is tissue culture or accessioning, but that's just my personal thing, but did it for years. So there's like different areas of the lab in one particular lab that you might enjoy doing over others, and just finding your niche. Maybe it's cancers or constitutional. This field gives you that.
Marcia?
So there are many things that I enjoy about my job. One definitely was Stephen said, the challenges that you get with the hardest cases. Because we diagnose and monitor patients, one thing that I find very rewarding is to follow these patients from birth until adulthood, and even throughout their whole life. And right now there are so many metabolic disorders that really can be treated, and they can lead to an almost normal life, and to me that's very, very rewarding. Other things that I like is the ability of being able to change and improve things and come up with new ideas. What type of workflow should we add this database to our workflow? Should we do that? So it's very, very exciting to me, coming up with new processes or new tests and new methods. I don't know what I like at least is probably trying to get everybody to get along.
That's a good one. I think we could all say that, no matter where we work. Raj, do you have anything to add?
Yeah, not much to add. It's just that it's like following, as Marcia said, and also Stephen, like, so getting interesting cases and solving them, it's always very, very satisfying. And also at NIH, you get to see the implications of your results pretty much in real time, such as bone marrow transplant, or because the treatment, most cases happens here. And also choosing who would be the best candidate for bone marrow transplant, and so on. And even if there is no diagnosis, you always, in this setting, you sit with genetic counselors and you see, and you can get a feel of how it affects the patient. So that is the most rewarding, I would think. And the challenging part is, and just following on Stephen's example, like, trying to find when you don't get any diagnosis, and it's particularly challenging at NIH because either the patients who are here have no diagnosis or already a known diagnosis. So sometimes you get a second diagnosis, and sometimes you look for research avenues, which could be, which is a kind of challenging, which kind of enjoy too, but it's most of the time that's more frustrating because it's kind of research. So you look for other causes. One of them would be this low variant fragment mosaic or somatic changes that's modifying the phenotype, or actually the cause of the phenotype. In certain cases that comes true, but most cases it don't. So the challenging part is developing essentially new pipelines, which is kind of it has its own rewards, but it's not as much as I like as giving the results to patients and making a difference.
All right. Another question from the chat. Do any of you currently have opportunities for fellowships within your own lab, or where would you recommend people look for such opportunities?
So I think in terms of opportunities for a fellowship, I think we do have a program, a fellowship program in clinical biochemical genetics. We have 2 position funded. So we do have every year the deadline application, I believe it's in September, and for next year. Right now, we just graduated 2 fellows and we have new fellows who have just started. So in terms of where can you find, I think the ABMGG would be a good resource for, you know, for a list of training program.
All right. Do people want to comment on how many cases they analyze in a day, just to give people kind of a flavor of the workload? And also feel free to comment on how else you spend your day if it is not all analyzing cases.
Just say I can't give a number. It depends. It's like but in general terms, it's basically divided between. For me, it's divided between doing research about 2-3 days a week, and research means like analyzing. So my research focus is the analysis of data. So of the genome data for undiagnosed patients or patients which we want to find more second or multiple diagnoses. So that's spent about 2-3 times, and the rest 2-3 days a week, and the rest is exomes and genomes, and mostly genomes these days, and whenever and CMAs. So and whenever we can fit that in, it depends on the need. There's no fixed number.
Carolyn, what about you?
So it's variable. Again, most of our cases, 2 technologists look at each case before it goes to the director. And so for a routine cytogenetics workup, for a constitutional case, a senior tech, normal, good quality, I can read it in about 30 minutes to an hour, my half. For a cancer case, I can read my half in just under an hour if it's normal, good quality. If it's abnormal, complex, I could spend the afternoon on it, just my half. For a normal array case, I can read it in about 30 minutes and write up the interpretation. If it's a normal known pathogenic case, like DeGeorge or Williams, I can probably read that in about an hour, pull the information together for the director. If it's of unclear clinical significance or it's not well documented, that could take me hours. So again, it depends on the type of case that I'm looking at and the quality and what's known about it.
All right, now we are running up in the last 5 minutes, so I'll just ask one final question, and it's one we've kind of touched on before, but it's clear that the audience is very interested in this, and maybe I'll pass it to Marzia first, because I think you are involved in your training program at your institution. But what kinds of things can people do in general to stand out as applicants to the fellowship program, regardless of what type they're applying to? What kind of things can enhance their application?
Well, I can answer for my program again. Besides the appropriate degree, would be having shown an interest in trying to understand what the discipline is. So someone who during their PhD has contacted the genetics program in their institution and has attended rounds with the geneticist, that definitely would show me that there is an interest in trying to figure out whether this is the appropriate path for them.
or if anyone else has ever been involved in reviewing these applications or having the application put in yourself, et cetera. Would you add any other tips just for people out there starting on their first round of applications?
No, as Marcia mentioned, and demonstration of an interest is absolutely necessary, if not critical. Like, that's.
Okay.
Take some initiative to show your interest in this field, that you've done more than just like, oh, I saw this was something I'm going to apply, to make a case as to, why you are a good fit for this.
Okay. And just because I don't know the answer to this, because I'm obviously not a laboratory director or technologist, but is there a written portion of this application? Do you have to do a personal statement, or is it just kind of a form, or?
I think there is a written statement. And also, from what I've been told from other people who are involved in application review, the letters of recommendation also seem to carry a pretty good weight. That I've heard that that's often a good deciding factor of who moves forward, of if they have really good letters of recommendation.
Right.
And it doesn't need to be necessarily someone who works in this exact field, but just, people that can speak to your, abilities and knowledge and ability to learn.
So choose wisely.
Yes.
All right, and we may have time for the last question. Does anyone have any experience with international certifications outside of the US that you want to comment on quickly? I don't know that anybody does, but.
I think I know that Canada has something similar to what we have. I am not sure in terms of laboratory director, laboratory fellowship. I'm not sure in other areas. I know that in Italy, for example, they don't have a laboratory fellowship, but the closest that they have would be a clinical chemistry fellowship, but it's not the same as a biochemical genetics fellowship. But Canada is probably the one that has a similar type of fellowship.
Is that your experience, Carolyn? I know you lived in Canada for a while.
So as a technologist, I worked here with my Canadian certification, and
okay.
my American more recently. And I don't even know that I had to, but that is how I moved down here, is on my Canadian certification. Sure. Yep.
Okay.
And, yep.
Good to know. All right, with that, I will just transition to our closing slides. So just as a reminder, next week at this same time, we will be speaking with physician clinicians. And again, even if you attended today's because you feel certain you're interested in laboratory genetics, give the other ones a shot, because you never know what interesting things you might hear about from other people. And then finally, the recording of today's panel will be posted on our website by Monday of next week. So if you have any friends or colleagues who couldn't make it, be sure to let them know. Please follow us on Twitter at ClinGen Resource for any announcements. And here is where you can find those recordings on our homepage. We will be sending you a survey after the completion of the series. Please take a moment to fill that out. Let us know how we did. We use that information to build this program for next year. So take a moment to do that. We would appreciate it. And that will go out on July the 26th. And with that, I believe that is our last slide. I just want to thank all of our panelists for giving us an hour of their time. It was wonderful to speak to you all and hear about your experiences. So thank you.
Thank you.